Okay, so let's get straight into it. If you've been at Berkeley for at least a week, or even from your own institutions, you may have received an email recently about increased incidences of influenza on campuses. The Centers for Disease Control has issued some policies that people should follow. So right now, if you didn't get your flu shot, you might be worrying that maybe that was a good thing to do 6 months ago. But these guidelines, I was just curious, are they informed by any sort of study for college campuses? And it turns out that they are. In 2011, there was this paper published in the Journal of Virology about the 2009 pandemic H1N1 influenza at UCSD. They wanted to understand how the flu was getting onto campus, how was it mixing, was it, were there clusters in particular dorms, were there clusters on campus versus off campus? And if you look in the methods section, they, amongst all the other technical details, they mentioned that they used a relaxed molecular clock and Bayesian skyline coalescent prior. And this word coalescent, this model is what I'm going to be talking about mostly for today, or for my lecture. You could say it's a basic model that underlies a lot of genetic analysis, sequence analysis these days. In these kinds of applications, they're used mostly for simulating genealogies, but the coalescent is actually an interesting mathematical model in itself, and so I'll talk more about that. So population genetics these days involves thinking forward and backwards in time. There's the Wright-Fisher model, which came first, and that was a forward in times model, in that you need a model for how generations follow other generations. But in the example I just gave you before, what the study was doing was sampling the student population and trying to understand, well, what processes gave rise to those samples, those observations. So you need to think both forwards and backwards in time. And the way we structured the 3 lectures is that I'll talk about the basic models and construct the coalescent, which is the basic backwards in time model. Anand, who's speaking next, will extend this model to more realistic situations. And then finally, Steve Evans will come and talk about the forward in time perspective, which is also very interesting, even though in a lot of papers these days you see mostly the coalescent perspective, applied literature. All right, so let's begin with the Wright-Fisher model, which was developed in the 1930s. The Wright-Fisher model is to, well, personally, I think the Wright-Fisher model is to population genetics what the simple symmetric random walk is to stochastic processes. It's the basic thing you need to understand the rest of the field. So even though it may not have that much relevance to a natural system, it is the natural starting point. So what we assume is that you have discrete non-overlapping generations, that there's a constant population size, capital N, and the basic idea is that individuals pick their parent uniformly at random from the previous generation. Well, this may seem a bit strange. Think about the even more basic alternative, which is you have a fixed population size and everyone just gives rise to one offspring every generation. That's even more boring. This is adding a bit more randomness, which is the previous, the next generation is composed of a multinomial sample from the previous generation. And Steve, who's going to talk more about forward in time processes, will give you a bit more of an explanation for why this might make sense. Right now, I'm just going to say that the probabilistic structure is that each generation is formed by sampling the previous generation. And for now, we're not considering types, we're not considering mutation, selection, or recombination. So here's a picture of the Wright-Fisher model forwards in time of a population of size 5. So there you have the first generation. And the second generation, the first individual picked the first individual from the previous generation as their parent. So did the second individual. The third individual picked the fourth. The fourth picked the fifth, and the fifth picked the fourth. So as you can see, not everyone in each generation makes offspring that have offspring that make it to the next generation. And so this is the process forward in time. Now, if we want to understand, given a sample, what may have given rise, what processes may have given rise to that sample, we want to understand what this process is backwards in time. So the way you do that is you pick a sample. So here we pick samples 4 individuals from the current generation, and we trace back their ancestry, so their family tree. And they actually all trace back to only one individual from the first generation. And for the remainder of this lecture, I'm going to focus on understanding this process. So we have a basic model for how generations are formed from the previous generation going forward in time, but we're really trying to understand what is the genealogy or the ancestral process when we take that forward in time process and run it backwards in time. Okay, so let me go back. So remember, the key point in the Wright-Fisher model is that individuals pick the parent uniformly at random from the previous generation. And now we want to know, well, when are individuals going to have a common ancestor? That's how we would get the ancestral process. So the first thing we want to know, let's just go line by line, is the probability that two individuals have distinct parents. And since each individual picks their parent uniformly at random from the previous generation, well, the first individual can pick any of the n individuals in the previous generation, so that's n over n, that's 1, and then the second individual just has to not pick the same parent, so that's 1 minus 1 over n. And once you go back one generation, you can repeat the process for k generations because it's the same thing. Once you go back one generation, they're still distinct. You do iterate. So the probability that two individuals have distinct ancestors for k generations is just 1 minus 1 over n to the power of k. So that's two individuals having distinct ancestors. Now let's generalize this to L individuals having distinct ancestors for k generations. So if we just focus on one generation, L individuals having distinct parents, you just generalize this for L individuals. So now the second guy can't pick the first parent, the third guy can't pick any of the first two, and so on. The Lth guy can't pick any of the first L minus 1. Yeah. And then for k generations, you just take it to the power of k. And I think this is the Kingman, who then went on to develop the standard coalescent, did this sort of calculation and saw that if you take population size n to infinity and you rescale time so that k generations, well, so you rescale time so your unit of time is n generations, which means one unit, k generations corresponds to k over n, that probability converges to e to the negative L minus 1 over 2 times t. And that corresponds to the time to coalescence when you have a sample of L individuals being exponentially distributed with this rate. And this is the key insight that led to the development of the coalescent. So now let me give you the mathematical construction of now let me first illustrate what this convergence means in figures. So here is your ancestral process for this 5 individual population. And then if you're taking n to infinity, you need to take the population size to larger, and you also have to take the number of generations, well, number of time steps larger. So that's what happens. And the idea is if you're only sampling 4 every time, this ancestral process is going to converge to this coalescent, which is you have these lineages and they'll coalesce at rate L minus 1 over 2 when you have L individuals in your sample. All right, so now let's look at the mathematical construction. And this was formalized by Kingman in 1982. I think there were several other people who were thinking along these lines, but the mathematical formulation happened now in 1982. And if you remember, the Wright-Fisher model was 1930, so there were 50 years in between when you had this forward in time model and when there was a formal description of the backwards in time process. So the n-coalescent is a continuous time stochastic process. It's given by capital pi n in square brackets, which is defined as just the set of integers from 1 to n. And it takes values in the space of partitions of n. And just to give you a feel for what that means, this is a graphical interpretation of the n-coalescent, or the 4 coalescent, but if you want to write it in terms of the space of partitions, you start from current time with the singleton partition, and then 1 and 2 merge, so then you represent that by putting 1 and 2 in the same set, and then 3 and 4 merge, and then finally the last 2 lineages merge, and then you've coalesced, you've gone to the single ancestor for the sample. So the coalescent process is described by the sequence of partitions and the waiting times between the transitions. More formally, you have an initial condition, which is the set of singletons. Transition rates are, you transition from one partition pi to another partition pi prime at rate 1 if pi prime is obtained by merging exactly 2 blocks of pi. And otherwise, the rate is 0. And this is the process until you get to the absorbing state. And just a few remarks. If you only look at the number of blocks over time, that's given by this hash pi n, that's a pure death process with a death rate at state L of that exponential rate. And as I said before, the sequence of partitions is independent of the number, how the blocks change, number of blocks change over time. So because of that, it's actually quite a nice process to study. And let me just take a minute to talk about some of the cute mathematical properties of the coalescent before I go more into the applications of the coalescent. So like I said, you have these two processes, a sequence of partitions and the waiting times between the mergers. So you can write down a closed form expression for the probability distribution of the Kingman's coalescent. Another interesting fact, probably interesting only to mathematicians, is that the coalescent comes down from infinity. So I described to you the n coalescent, which is you start with n individuals and you look at the ancestry back in time. It actually makes mathematical sense to define the coalescent starting from all the positive integers. So that's the infinity part. That's called dust. But if you run it for any positive amount of time, it becomes a partition with finite numbers. So it comes down from infinity. Not all coalescents do that. Kingman coalescent does. And related to...
Did you define what other coalescents are?
I haven't yet, but this is the Kingman coalescent.
But you could just say what you mean by other coalescents, like explain how they...
Other ancestral processes that don't go this way. So this is the Kingman, this is the description of the Kingman's coalescent. You don't have to make moves this way, right? And so those I just refer to as other coalescents.
So you could merge more blocks.
Mm-hmm. Yep, and I'll get to that later. So related to this concept of coming down from infinity is this interesting fact that if you project the n coalescent to a smaller set of integers, you get, so say m integers, you get an m coalescent. And this actually is very obvious when you think about it in terms of an ancestral process. If you have an ancestral process for 10 individuals and you're not interested in the 10th individual anymore, you still have an ancestral process for 9 individuals. But from the perspective of a stochastic process, what you're doing is taking an n-dimensional, something like in n dimensions, and you're projecting into a lower dimensional space, and it still preserves the same properties. And that is quite surprising. Okay, so these Q properties aside, why is it that we study the coalescent? I've given you the derivation of the coalescent from the Wright-Fisher model, which is, although a basic model, is just one model of how populations can change over time. As it turns out, the coalescent is the limiting process for a large family of reproduction models. And to characterize this large family, I need to define the Canning's model. So the Canning's model is defined by these vectors, nu sub 1 through n, and is still our population size. And the mathematical characterization is just that these nu sub I are exchangeable integer valued random variables and they have to sum to n. Exchangeable in an intuitive sense just means you can relabel these nus and you still get the same probability distributions for all your calculations. So they're sort of, it doesn't matter how you label your individuals, your results are always going to be the same. The biological interpretation is that nu sub I is the number of offspring left by individual I from the previous generation. So in the Wright-Fisher model, these nu sub I are multinomial with probability 1 over n. But you can see that there are many other possibilities for how you define the specter of offspring. So Merle proved in 2000 that, let me just preface this with, so not all Canning's models will give you Kingman's coalescent. And Merle proved that if this limit is true in a Canning's model, then the genealogy of the sample population converges to the Kingman's coalescent. So what does this fraction mean? Well, the numerator is the rate, is approximately, you can think of morally, the probability that 3 blocks merge into 1 block. And the denominator is the probability that 2 blocks merge into 1 block. So all this is saying is that the rate at which 3 merges occur vanishes relative to the rate at which 2 merges occur. And that makes sense because Kingman's coalescent only really, I didn't specify this, but it only allows binary merges. And you can imagine if you have a population model given by a Canning's model where 3 merges are possible and on the same order of magnitude as 2 merges, then you wouldn't get Kingman's coalescent because you'd expect 3 merges and Kingman's coalescent doesn't have them. So that's why Kingman's coalescent is studied quite a lot, because it has this universality.
Should be again the small n.
Yes. Sorry. I changed notation halfway through my presentation. All right. We're back to begin. So far I've told you about this ancestral process of the Wright-Fisher model, and I've told you that the coalescent is the limit of that ancestral process, and that is also the limit of of that Wright-Fisher model, and it's also the limiting process for a lot of other models. But I haven't talked about types or mutation or anything like that. I've only talked about what is the family tree of the sample. So now let's suppose that we're interested in the types of the individuals, not just their genealogy. And the nice thing about the coalescent, if you have no selection, is that mutation can just be a Poisson process on top of this tree. So there's the tree, which is our coalescent, and then we drop mutations at a Poisson rate of theta over 2 onto this tree. I'll explain why we divide theta over 2 on the next slide. Now, the interpretation of what the result of this process is depends on what assumptions you're making about mutations. So 2 common mutation models are the infinite alleles model and the infinite sites model. And the infinite alleles model assumes that each mutation leads to a distinct allele. So every time there's a mutation, you have a new type. So if you are numbering your types by the integers, every time there's a new mutation, you increase the integer that you're labeling your types with. And the motivation for this kind of assumption, I think, was because before sequencing, the data that biologists collected were of the form of these allelic partitions, where A sub 1 equals 18 means there were 18 types that only had one individual representing them. A sub 2 equals 3 meant that there were 3 types that had 2 individuals representing that type. And here, A sub 32 equals 1 means there was 1 type or 1 allele where there were 32 representatives in the population, in the sample. And they wanted to understand, well, is this typical? What can we say about this allelic partition? And this was touched on in Monty's talk, which was you can calculate the distribution on these allelic partitions using U sampling formula. U sampling formula came before Kingman's coalescent, but you can use Kingman's coalescent to derive, give another derivation of U sampling formula. And I put this up mainly because you can see here theta appears nicely this way, so that's why typically you say the mutation process is Poisson rate theta over 2. So that's infinite alleles, and now that we have more sequence data, infinite sites seems to be more relevant. So here you assume that each mutation occurs on a distinct site. So before, each mutation led to a new type. Now we assume we actually do have a sequence, and that every time a mutation occurs, because there are so many sites, the probability that you hit the same site is so low that every mutation just occurs on a new site. And again, you drop mutations on like a Poisson process, but now we're going to just label them because the way we're going to do the accounting requires this labeling. And whereas before you had the allelic partition, here it's not so clear. Like if you have a new site and you have a mutation on a new site, what object to look at. So to illustrate this, what I've done is on this table you have all the different mutations, and these are the individuals. So individuals 1 and 2 just experience one mutation at site 3. So 1 and 2 have a 1 here. That means they have a mutant at that site. 3 and 4, well, 3 had 2 mutations on 1 and 2, and then 4 had 3 mutations. So you have this kind of data that's generated from the coalescent with mutation, assuming an infinite sites model. And here the thing that we're interested in looking at is called the psi frequency spectrum, which is for a sample of size n. I think I was supposed to have lowercase n. Sample size, no, we can talk about that. For a sample of size n, psi n I is the number of sites at which exactly I individuals carry a mutation. So, for example, here site 3 has 2 individuals carrying 1 mutation, and site 4 has 1 individual carrying 1 mutation. So this is the kind of statistic you look at when you assume an infinite sites model. So mutation is, usually it's infinite sites and infinite alleles, and the kind of work is done based on psi frequency spectrum or the allelic partition. And there's more theory that can be done to calculate statistics about those 2 objects. So moving on from the basic Kingman's coalescent and mutation, I want to talk about when Kingman's coalescent is not appropriate and what kind of other coalescents might be relevant. So the Kingman's coalescent is not appropriate for for many reasons. Here are 3. One is if there's a population bottleneck. So if you have a new population that's seeded by a very small population, then if you went backwards in time, you would expect a lot of individuals to have the same ancestor very quickly. Similarly, you can have a selective sweep from beneficial mutations. So if there's a mutation, say in bacteria, that confers antibiotic resistance, and then you apply antibiotics to this colony of bacteria, then only a few bacteria survive. The rest of the they fill up the rest of the population. And so if you look at this process back in time again, you'd expect a lot of coalescing events. And finally, large variability in offspring distribution can also give rise to situations where the Kingman's coalescent is not appropriate. The main example in this is marine populations, and specifically oysters. So apparently oysters are very widespread spatially, but the genetic variation from generation to generation is quite low, which leads people to believe that each new generation of oysters is founded by just a very small number of oysters from the previous generation. And again, if that's the case, if you're running the process backwards in time, you'd expect a lot of individuals to have the same ancestor very sooner than you would expect on the Kingman's coalescent. Yeah?
Do you have any thoughts on how that kind of relates to using that to look at disease outbreaks? If you consider like a transmission process as being a population bottleneck, for instance, do you think it's an appropriate application of the model?
It might be. I know that there's some work on HIV in that direction, but I don't think it's an area. I'm not familiar with that area of research. I haven't come across it. So this leads us to look for other coalescents, and now I will define what I mean by other coalescents. So the lambda coalescent is actually just a subset of the other coalescents you can have, but it allows for 2 blocks to coalesce into 1 block. You can have multiple coalescents to multiple blocks, but the lambda coalescent only allows multiple coalescents to 1 block. And the lambda coalescent is specified by this lowercase lambda bk, which is the rate at which a particular k-tuple of blocks out of b blocks merges. And the reason why it's a capital lambda coalescent is because Pittman in 1999 showed that the coalescent process is associated with this lambda can be uniquely characterized by this finite measure capital lambda. And it's because of this characterization that these processes are called the lambda coalescent. So let me just go back to my analogy with stochastic processes. I said Wright-Fisher model is like the simple symmetric random walk for stochastic processes and population genetics. Kingman's coalescent is like Brownian motion. A lot of things converge to it, but not everything converges to it. And the lambda coalescent is like Levy processes. If people familiar with that area, they might understand. I think it's useful to have that analogy. There was a question?
Yeah, can you explain what a block is?
Oh, yeah, right. Better if I just show you the picture. So these are blocks.
Okay.
These are the sets. They're also called lineages. So the lambda sub kb is just the rate at which these blocks converge, merge into single blocks. All right, so
Oh, wait, just go back one slide. So the X there in that formula is kind of like the probability that there's an event that causes K blocks to converge to B.
you're thinking of it as a binomial, the
Yeah,
P in the
yeah.
binomial distribution. It's related to that. So if you take this x to the -2 and put it under this lambda, then this is the binomial probability, and then this measure has another interpretation. So it's not so x in that context, yes, but then how do you interpret it in the broader context is not as clear. It is not so straightforward. All right, so I've talked about the basic building block Wright-Fisher model. Steve Evans will go back and talk more about that. The Kingman's coalescent, the standard vanilla coalescent, how you put mutation onto the coalescent, and then in these certain circumstances when you can have multiple blocks merging, what might be a reasonable approximation to that, the lambda coalescent. I haven't mentioned recombination, I haven't mentioned selection, I haven't mentioned population sizes changing. So in the next 2 lectures, I hope you'll come back after lunch. Anand will talk about how the coalescent can handle recombination, what happens when the population size isn't constant, which is often the case. And I've only talked about the backwards in time perspective, but there are many advantages for using a forwards in time perspective. In particular, you can handle selection more easily, I think. So that's what's coming next. And for those who are interested in references, these are just a few that I found helpful to get some background on this topic. Okay, that's it.
We'll take time for questions.
So what do people use these for?
For simulating genealogies. So I'll just talk. In that first paper I showed you, they wanted to understand how, we took samples from all these students, well, what may have given rise to them. A lot of the times they'll build a phylogenetic tree from the samples. But to understand whether this tree is typical or atypical, to find out what distribution the tree comes from, you need to have a concept of what is a random tree. And I think a coalescent in those situations is commonly used as a way to simulate a random genealogy. Does that answer your question?
I think so, yeah. You got data and you first build a tree from that and then you...
So yeah, originally it was done that way. Now they're a software like Beast, and what they'll do is they'll take a coalescent as the prior, and then given this data, they'll calculate a posterior. So it underlies a lot of the analysis that's done. And if other people have other uses for the coalescent in applications, feel free to chime in. But it's at the very basic level, coalescents are being used to generate trees. You wouldn't actually, I don't think you'd manually do that now. I think you would use the software.
You also get a lot of exact formulas, right?
From the call, yeah.
For
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 the,
whatever you're interested in, right? Distribution of allele partition, etc., under a neutral theory.
so say you're collecting data and you can match it to the site frequency spectrum. Assuming the coalescent, you can calculate statistics on the site frequency spectrum.
So I would have some site frequency spectrum from data, and then I would look to see if that showed up in my coalescent model.
By varying parameters. I think that's one way.
Can you say just a word or two about how Beast works, or how maybe other things that fit these models work?
I wish I could, but I'm not as familiar with the software. I know it exists and I know that they have coalescent priors, but that's the extent of my knowledge.
How does the behavior change as you sort of throw in the fact that for 2 parents, let's say, sort of that kind of graphical?
You have 2 parents, so if you have a diploid population,
Yeah, I mean, just somehow this.
okay. I think the most basic thing to do when you have 2 parents is you assume that each individual has 2 copies of their genes. So instead of n, you have 2n, and you still think of the process going back in time as, you know, one of your, I'm going to use the wrong terminology, one half. So you just, each individual is now two individuals under that haploid. If you want to include recombination, which is a more complex issue, then Anand will tell you in the next lecture.
But the nice thing is, yeah, even if individuals have two parents, each allele that individual has just comes from one parent. So it's not like the two parents get like perfectly mixed together or anything.
I think I missed something. Can you please explain again why the lambda coalescent is better suited for non-neutral models?
Oh, right. So I have to be careful in my terminology. It's not so much that it's better suited for non-neutral models, because if the selection is not very strong, it's still not, I don't think it's still a very good model. But when you have such strong selection that a large fraction of your next generation comes from one individual, then that corresponds to having multiple blocks coalescing backwards in time. So in that situation, it might be that a lambda coalescent models the process well. But the lambda coalescent, in its assumption, because you still have, I forgot to mention, you still need exchangeability. Exchangeability sort of implies that each individual is sort of the same relative to the other individuals. It isn't consistent with having selection acting on the population.
So with lambda coalescent, because you have multiple coalescent events occurring in a short period of time, you have better estimates of time to the most recent common ancestor, for example, but you're still stuck to the bottleneck or the selective solution, correct? Yeah.
If there are no more questions, maybe we can take one
Right.